Hello everyone, my name is Bo Bush, President and Founder of Osmosi. Today we'll be going over the improving of our predictions and competitive intelligence in the clinical trial landscape using COVID-19 as a backdrop for a case study. First, maybe a little bit about our company. Osmosi has been providing consulting services in the pharmaceutical industry for 8 years now and data services for the last 4. We've recently entered the marketplace for investment firms, where our data seems to be very popular due to how clean it is and some of the predictive capabilities, which we're going to speak to in a little bit. But our data services really helped inform our consulting and made our consulting stronger. And likewise, our consulting has really informed our data services and how our data could be cleaned further to automate the modeling outcomes as well as the reporting outcomes. Those really are some important ingredients for what we want to discuss here today. It seems like the past few years we've been discussing natural language processing techniques, machine learning, artificial intelligence, and it feels like something we've all been looking at, something just a little bit out of reach for a lot of us. We hear other people are doing it, but we're not really seeing, real meaningful or tangible results. And really that's the point of what we want to discuss today, and bringing some of the fundamentals that are going to be needed in order to move forward with that. Fundamentals that we all actually have at our fingertips now. So let's get started. So first, let's talk about the predictive models, right? We have data today. Everybody has the best data that they want or that they're using today. We have ours. Everyone has theirs. But quite often we're drawn into thinking about it just in terms of the way it exists today. What we're suggesting is we take that same data and we have a view of that same clinical trial, for example, as it existed yesterday as well, and maybe even last week or prior to that. And when we have that, we can start to see directionality, we can start to pick up on trends, we can even look at how all of that flows for similar trials within that company, within that disease area. And we'll show an example of that in the following slide, but this is probably the first place to start. It's the lowest hanging fruit where you can really get started in starting to make some strong predictions. And we'll give you an example of that in a minute as well. Next, data standardization. This is not sexy, guys. I get it. And so how can you take a large chunk of text and extract specific categories or highlight certain flags to assign to that data element? But this is the key right here. Once we are able to nail this down, once we're able to build the libraries that are specific to our business needs, these things start to pour out information and abilities to categorize information that will further help us predict the kind of trends we see when we have that time series approach to the data that I just mentioned in the predictive models. And we do this already, right? We're able to extract endpoints out of the text-heavy descriptions, or endpoint timing, or mechanism of action. Maybe we'll pull that from a different data element, PubMed or something. The drug names, how can we assign that non-proprietary name to all those complex drug names? And then finally, what it comes down to is that library maintenance. Now, the standard way is a more human-powered, heavy work approach, and what we're going to suggest is more code-based training for our libraries, and still using that human element, but in a much more efficient, effective way. And I'll describe that full circuit library maintenance in a couple of slides. When we have that, we're able to link our data based on those categories to other more text-heavy, text-rich information that will bring its own categories or information alongside it, right? So even things as simple as clinical facilities and their locations, and the ability to put footprints on the clinical facilities that are associated with different disease areas or frankly different companies. And we can look at launch strategies along those lines, right? We can start to predict these in advance for different companies. Then we can think about even clinical results or adverse events. How can we link those to a product as opposed to just a trial? How can we roll up all of the results or events around a certain product across all of the trials to a certain one that we're looking at more specifically? And of course, this would even expand the PubMed abstracts, drug labels, FDA approvals, a whole host of other things once you have that data standardization in place. And enriching your data that way and even further categorizing it or tagging it that way help to support the predictive models that I mentioned up above by giving more characterization, more classifiers to your predictive models. Okay, so let's talk about the time dimension component of the data that I mentioned earlier. What we have here is a clinical trial with a few data elements. And it was last updated on April 6, 2020. We used some of those data standardization approaches and enrichment to clean the drug name, to designate whether or not that drug was novel or not, the treatments, the disease area, that sort of thing. This is all very helpful when you're trying to do some reporting or some nowcasting. But what if we unpack that trial? What if instead of looking at that last version, we thought about them all since February 1st, 2018, when it first showed up? What if we thought about all the different elements and all the different edits that happened along that time? You really get to see the recruitment timing and how that baked in. You get to see how when recruitment was finished, it appears that they recruited faster than they thought they might have, and therefore as soon as they finished recruiting, they were actually able to put in a more accurate primary completion date. Totally makes sense. What we're seeing because of the data is how important that recruitment time is, obviously, and it's one of the reasons why we started adding percent accrual into our data, and we're just starting with what the original target was, and then we're overlaying the actual so that you can see how far along you are. And we're finding we can actually make predictions about primary completion date based on this information. Most times more effect or more closely than the primary sponsors themselves. In fact, when a trial starts, we're typically right, or we typically predict the primary completion date more accurately than the primary sponsor, 2 to 1. Look, we know why that is. We're using just the straight data. We're able to analyze and make predictions based on the company and their history and the disease areas. But the primary sponsors, they typically have more pressures than just the modeling at play when they're trying to put together a primary completion date. But it's little small things like this when they add up and get a little bit better at making predictions based on the data at hand. Okay, next let's talk about data standardization and a must have your own pharma-focused natural language processing libraries. This is the key, guys. Anybody who's still cleaning data with a huge army of experts to clean the data, they're still not going to be able to deliver it in the consistent manner that code-based approach can do. But more importantly, they're losing in the ultimate race of developing the most comprehensive libraries, because those libraries will be the key to moving into the next steps of machine learning, artificial intelligence. And if you're still relying on a huge army to do all the cleaning, there is a role for that, for the experts, but you don't need that big army of folks doing it. I'm going to walk you through an example of this, and finally to that full circuit library maintenance, which is absolutely the key to winning that race to having the most comprehensive library. So let's just start in general. Here's a bunch of raw names of drugs in clinical trials that we're tasked with cleaning every night when our clinical trial data comes in before we push it out the next morning to our clients. So the first thing we need to go through is go through each of these, find the aliases, and assign the appropriate non-proprietary name. And from that, then we can further tag it to things like mechanism of action. We can even further tag it to, has this been approved by the FDA, or some of the other data elements that are driven off of product names or mechanisms of action. I'm not going to go into details on that. I think you guys get how all of that is connected. What I want to speak to now is the first half of the circuit that a lot of folks are getting right now, and that is when it comes in, the new data element, the first question is this in our library, yes or no? And if it is, just like I described, it's fairly straightforward. You find the right alias, you link it to your non-proprietary term, and then you're able to output that out as the correct answer or the cleaned version for that drug. Okay, now let's talk about when that search term that you're looking to clean is not in your library. It's a new drug perhaps, or a new version, or a new alias. How do we complete the circuit, the other half of the circuit that we just described? And this really is the key in winning that race to that more comprehensive pharmaceutical focused library. We have a whole slew of publicly available text-heavy information out there for finding these. And so we can search through that. PubMed gives us a backdoor into it. You can look at BioBert. That's another example of Google kind of technology to be based on PubMed articles. We've explored that a little bit ourselves. We found another route that was better for us. But this is going to be the key, right? We do that. Now look, we could have a human simply type something in and search through it and find the right item, right? But what we're looking to do is where that's easy, where that's best, have the machine, have the computer, have the algorithm take care of that. And what we spend time is having humans maybe check the results and understand the confidence intervals with which the results are the right answer. And over time, we essentially continue to train our algorithm using these check-ins to get better and better. And when you have that, that's the right use. If we're going to have our experts doing the mundane and the cleaning, why have them clean every single iteration when really what they could be doing is approving the outputs from the code that's already been written? And when you have that and you have a confidence interval essentially assigned to every version, you'll learn and you'll be able to be trained almost by your algorithm what the outputs are that you can trust to just send directly to the output. And when it's new, you can also use that to update your library with that new alias, that new term, so that the next time it does come through, you already have it in your library and you can go down the first half of that circuit. That's really what I meant earlier when I was speaking about the full circuit library maintenance. This is something that evolves over time and gets better and better, and it needs to continue to evolve and grow as new technologies come on, and they come in fits and spurts, right? It's not like a steady, even pace. So you couldn't even hire for it appropriately in terms of the ups and downs, but the PubMed, the scientific knowledge will be there. It will be in place typically long before we see it in clinic. Okay, so let's talk about some ways in which we can apply this in our day-to-day activities using COVID-19 as a case study or as a bit of a backdrop. So here's a standardized kind of view. This is for treatments in clinic for COVID-19, the non-vaccine type treatments, and just applying a couple of categorical types in the bullseye chart, or the pie chart that you see on the right, and just really breaking drugs into categories for explaining in terms of their approach. So we could see that roughly half the trials are really treating that anti-inflammatory approach, are taking that anti-inflammatory approach, really treating the symptoms of COVID-19, which is fantastic. And then the other half of the trials are focused more on the antiviral activity, really going after the virus itself, and a few of those kind of both. And really when we can look at trials. And again, that's that standardization. How can we look at text heavy and then assign a category based on that? These are examples of how we can take something that's important to us in terms of a reporting output or something along those lines and tag things as they flow through. Same thing on the other side, on the left-hand side, where we just look at the number of companies or the treatments. If we're not doing that cleaning, we can't actually automate the summary of these elements because we never know if we're double counting certain things. Same thing with the facilities, and as we get better and better with facilities and the patients, that's going to be really helpful in understanding recruitment, competition for same patient groups within the same clinics, and that sort of thing. And we've got an example of that coming up. When we have these categories easily in place and automated, you can then start to think about things in terms of other dimensions. In this example, we're thinking about the patients rather than the trials that fall into the different treatment categories, where before it was kind of 50-50 between anti-inflammatory and antiviral. You can see that the majority of the patients are actually falling on the antiviral side of things. And again, all of this cleaning, you could start to turn from a dimension of companies or the clinical facilities and clinical facilities patients, that sort of thing, and start to make estimates along those lines. So let's walk you through some of these estimates using the COVID-19 vaccine clinical trial space. So the headline here is there are a number of vaccines in clinic right now. We're all aware of this, and what it means is there's a high likelihood that next year we will have the option, not just one vaccine, but multiple vaccines. Which vaccines will people take? Which ones will be available? And so we're going to walk you through our predictions in recruitment and some emerging strategies along those lines. So the flag chart you're looking at here represent clinical trial starts this year for vaccines in COVID-19. First one to start a clinical trial, Pfizer and BioNTech, in late April, going for roughly 30,000 patients. Now, by vaccine standards, 30,000 patients is a little bit light, but given the environment, it made sense. Moderna followed suit with 30,000. Clearly, there's some guidance along these lines. AstraZeneca shortly after with their 30,000. Late to the game, Johnson & Johnson falling behind by 3, and yet they show up with a clinical trial for 60,000 patients that they're looking to build. And then CanSino comes along with 40,000. Now, what's interesting to me is that when we speak to people about their likelihood of taking a vaccine, if there's one available by the end of 2020, most frequently they respond with they'd be interested, but they would wait for a little while until more people had taken it and there weren't any issues. And that's when we started talking to them. Did you know that by the end of the year, we think that more than 100,000 people have taken a vaccine? That changes the game quite a bit, and it changes their answer quite a bit. And they're looking towards, if that many, maybe they would go ahead and move forward. I'll show you how we got to these answers. When we look at the COVID-19 vaccine clinical trials through the lens of the clinical facilities, we see that there are roughly 240 of these covering all but 3 of the United States. And when we have that perspective, each of the blue dots is where a clinical trial for a vaccine is taking place. And the larger the blue dot, the more clinical trials that are happening in those facilities, so the more competition for those patients. When we have all of that at our fingertips, when we have that point in time view of the data, it gives us all the pieces of information for the equation for predicting recruitment. And that's how we landed on the total of over 130,000 people or patients by the end of the year receiving at least one dose of a vaccine for COVID-19. At that point in time when we made this prediction, Pfizer's target was just under 30,000 patients. They've since increased to 40,000, which I think is pretty smart because there's a strategy that's emerging here. But before I get to that strategy piece in the next slide, this is another example when you can take the information you have, but because you have it geolocated, you can also overlay other elements, right, that support your recruitment or support whatever the item is you're trying to solve for. So in this case, we overlaid the shading of each of the states based on the incidence of COVID-19 per 100,000 population. This comes from CDC, and it's state-based or zip code-based that you can align that with your clinical trial data. So just another example of how when we're able to tag or code things in one way, it allows us to bring in additional data elements to further enrich those predictions. But let me get you back, let's get back to the emergent strategies here. So when we're looking at our recruitment predictions by month, by clinical trial, a few things pop. We see J&J really emerging. That's where we're saying they were going to be the leader in the number of patients that they had on vaccine by the end of the year. Now, this data was as of the beginning of the month, which in COVID-19 terms is practically ancient. So much changes day to day in this landscape. And I was actually tempted to update this with the latest and current, most current data, but then I thought it best to show you what we predicted at the beginning of the month and speak to how well has that played out. And Pfizer's a great example of this. Pfizer, we predicted by October that they would have achieved their target number of patients. In fact, they actually did that about a week ago or so. They already achieved that. So we were right on the October number, and since then they've increased to 40,000, which when you look at this chart absolutely makes a lot of sense, especially when we're starting to hear concerns about people saying, well, I'd love to see more patients take this before maybe I would show up and get a vaccine once these vaccines make it to the market. We still believe J&J's got a great strategy of going for the most patients. I think Pfizer does too. They increased to 40,000. I would not be surprised if they don't choose to increase that even further as they move along. That would really put them at a strong advantage with J&J. And again, AstraZeneca, we hope to see those guys to be able to get back on track. We were predicting at the time that I think right as we made this prediction, we were hearing that they might be on hold. If that hold holds out much longer, I could see us adjusting our estimates. But this is a perfect example of really the culmination of when you have everything at your fingertips to be able to make predictions and use machine learning with really what we have today with just some simple standardization, point in time reference of data, not just the most current, and then you're able to overlay other elements with it. Okay, so bringing us home and wrapping this up, that state-of-the-art competitive intelligence is in reach today. People are achieving it today. Some common trends that we're seeing in clients that are getting there faster than others. Number one, they're automating the mundane. They have competitive intelligence experts with literally decades of experience, and they're either spending the majority of their time in data processing or moving little squares around in PowerPoint, or they're spending the majority of their budget paying vendors like me to do that. And what we're trying to do is help them to automate that and streamline that through our reporting tools. And we're seeing those that are really moving faster to doing that, and getting their experts out of that business, getting their budgets out of that business. They're establishing a single source of truth. That's another element that they're doing. It's really back to automating the mundane, because they're building rules for sharing information across their teams and even across cross-functional disciplines to support information in a single source of truth that provides the reporting. So they're spending less time cross-referencing, why did you send that prediction or that estimate to senior management and I sent this different one, and who's more accurate, right? And who should be reporting that information? They sort that out in advance, push it all to a single source of truth with some automated reporting outputs so that senior management can stay focused on getting the information they need more timely, and the teams can spend less time cross-checking why different information was sent. And what that allows them to do is focus again on the implications of the information rather than cross-checking, data processing, moving squares around in PowerPoint. And finally, an increased focus on clinical facilities and the strategies that are emerging from there. This is going to be incredibly important in thinking about recruitment predictions, even likelihood of success, I would argue, and definitely in terms of forecast uptake. How many treatment professionals already have experience with this product from the clinic, right? So those are just a few examples of what we're seeing, common trends from clients that are really moving along these lines faster than others. And I want to thank you all for the opportunity to present today. I'm really looking forward to the conversations that come immediately following this presentation, as well as in the future along these lines. And finally, I also want to just thank Pharma CI for thinking out of the box and holding this virtual conference for us. Thank you very much.